This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 47 for August 28th, 2009. Hey everyone, it's Vincent Racaniello, and guess who's with me?
Dick Despommier.
It's Dick and I in my office. Cloudy, rainy day here in New York City.
Absolutely.
But it keeps the mosquitoes away.
At
least the West Nile mosquitoes prefer to eat birds.
Correct. This has been a cold, wet summer, and we've had 2 cases of West Nile east of the Mississippi almost. I mean, it's been remarkably predictable in terms of the outbreak.
Yep. And it's very interesting that about a year ago, Dick and I sat down and had lunch.
Yep. And
decided to start this podcast. The first episode was about West Nile virus.
Yeah.
And we can't get away from it, not that we want to.
No, it's still with us.
It's a good story. The whole thing is great, and Dick, of course, wrote a book. Dick, in case you're wondering, Dick is a professor here at Columbia in the School of Public Health.
I would like to correct you on that, Vince.
You're not a professor?
As of January 1st, I received my letter from the president of Columbia University announcing to me the fact that I have been elected emeritus professor.
So you're now an emeritus professor?
I am.
As we sit here in my office, you're emeritus professor.
As we sit here.
Health. Well, Dick, to me, you'll always be professor.
I'm deeply honored to be an emeritus.
What is it? Let's tell people what emeritus means.
It means you're an old fart.
Okay. Very good.
That's what it means. No, here's what I think it means. And I don't mean to belittle the honor. It's an honor. It's not an expected outcome. Okay, so at least this is what I'm told. I'm told that only professors at the university are considered for emeritus status, and in order to get it, you have to be nominated, and it has to go by the board of trustees and the president, and it has to be, I think, unanimous. And apparently mine was, so
I'm not surprised.
I'm deeply honored, though. It's still a big honor.
Now, I think what it means is that you can always call yourself professor.
Right. But it also means you've reached a certain stage in your life, too. Because I remember when I was appointed at Columbia in 1971. I remember when I got my tenure in 1986. I remember all these landmarks in my professional career. So to have a letter that says you're now an emeritus professor is sort of a life-changing event.
Of course. But you're doing other things, and you will continue to do other things.
Absolutely. No.
You'll continue to be here at Columbia, I understand.
I will.
You'll have an office, and I
have an office.
I think you will continue TWiV.
Absolutely. Why would I ever not? I have such a wonderful time with you, Vince. And with the audience, I must say.
The audience is wonderful. In fact, today we're going to hear a lot from the audience. We have tons of email, and they're fabulous, and they drive this whole episode. But before we do, I want to talk about a piece of writing of Dick's that I read this week in the New York Times. It's called A Farm on Every Floor. It's an op-ed contribution. Congratulations, Dick.
Thank you, Vince.
Can I read the first paragraph? Because
Sure.
It's beautiful.
Please.
If climate change and population growth progress at their current pace, in roughly 50 years, farming as we know it will no longer exist. This means that the majority of people could be soon without enough food or water. But there is a solution that is surprisingly within reach. Move most farming into cities and grow crops in tall, specially constructed buildings. It's called vertical farming. This is a perfect introductory paragraph. You set up the problem and the solution, and you pull people in. Why did you write this, Dick?
Because I was asked to.
You were asked to. What an honor.
It's another honor. Exactly right. I was knocked out by the piece. I don't agree exactly with the drawing that comes with it. It's got a little propeller on the top. And
it's an image of a skyscraper with sun shining on it.
Yeah, the sun is okay. It also collects rainwater. That's good, too.
Water flowing through and going into some kind of box underneath it.
Right. Probably a collecting device of some
sort. What's the propeller for?
I think that's supposed to be a wind turbine.
Is that what you envision in this vertical farm?
I envision collecting wind power, but that's not the way to do it.
I see.
But they've also put cows at the bottom. I don't think that we're going to have 4-legged critters swarming around this vertical farm, to be honest with you.
Oh, they took the cows out of this.
Did they?
Yeah, unless
that's good.
But I understand that in your article you talk about them being within the building, right?
Not cows.
Sheep.
Did I say that?
Goats.
I didn't say that.
All right, some kind of animal? Humans, yeah.
Well, definitely. But I think you can have chickens and you can have aquaculture of various sorts, shrimp and mollusks.
Well, I think everyone should read this. It's a great article. It's a great idea, which you've heard of over the year that we've been doing this podcast from Dick. I'll just close with the last paragraph. When people ask me why the world still does not have a single vertical farm, I just raise my eyebrows and shrug my shoulders. Can you do that here in the office?
Sure.
He doesn't know. Do you think we will have a vertical farm soon?
I do, actually. I just got back from a meeting with the mayor of a very large eastern city, but not the one I work in.
Mm-hmm.
And I was asked not to mention this yet. And this particular city is extremely interested in developing this concept into reality.
Excellent.
So I think the answer is yes.
Well, Dick, when it's built and functioning, maybe we'll do a TWiV from that first vertical farm.
That'd be great.
That would be fun.
That'd be wonderful.
We have a story actually today that ties into this. Not too long ago, a paper was published saying that you could make a vaccine against influenza virus using what's called virus-like particles. So what's done in this paper, which was published in PLOS 1, the title is Protective Immune Responses to Influenza Viruses with H5N1, NA, and HA elicited by an influenza virus-like particle. So what they do is they take the genes encoding influenza virus, HA, and NA, and they express them in insect cells using a insect virus, a baculovirus. They take the HA, the NA, and the M1 genes of influenza virus, and you express them in insect cells, and the cells make particles. They bud from the cells. We haven't talked about budding, but we will in our Virology 101 course.
Exactly.
But influenza viruses are produced at the cell surface when particles bud off. They kind of pinch off. But anyway, they produce these particles in insect cells, and they're immunogenic, and they protect ferrets and mice from infection. So that's very nice, because these are not infectious. They have no genomes in them.
This creates a sterile immunity, Vince?
Sterile meaning?
No infection.
No infection, exactly.
That's a
The
sterile
particles
infection.
are just there.
It's amazing.
And they're immunogenic. And they're easy to make. They're not infectious. They're not biohazardous. So it's a great system. And in fact, this whole concept was shown years ago in mammalian cells. You could express flu genes. In fact, Dick, this is very interesting. You actually don't need the M protein. And you don't even need the NA. If you just express HA, the hemagglutinin of influenza, in cells, you get particles made. They stick to the surface of the cell because of the sialic acid binding the virus. And the neuraminidase, if you express, then it allows particles to be produced. But M protein isn't needed, apparently. And a virologist by the name of Robert Lamb at Northwestern showed that a number of years ago. So anyway, this is quite interesting. I think there's a big future for this in vaccinology in general. Because you could do this very rapidly. Starting from a sequence of the HA, you could make particles quickly.
Right.
So I blogged about this, and in the comments section to that post, Ed, who has a blog in South Africa called Viral Bloggy, he wrote, "Did you know you could do this in plants?"
Really?
So I looked it up. I had heard of this, but I'd never looked into it. Because in plants, the membrane of the cell of plants is right next to the cell wall.
Right.
So you could imagine this would be difficult to do. But it turns out that a number of virus-like particles have been made in plants. Hepatitis B is one of them. But those are produced inside the cell.
Sure. I remember something about bananas. They were incorporating that into
banana.
Yeah,
So you eat the banana?
And you get vaccinated at the same time.
So I looked this up. This is fascinating. And here is a paper, influenza virus-like particles produced by expression in Nicotiana benthamiana, the tobacco.
Finally, a good use for tobacco plants.
Induce a protective immune response against a lethal virus challenge in mice. So what they do here is they put the gene just for the viral HA into the plant cells, and they get particles made. They're produced at the plasma membrane, and they sit between the plasma membrane and the cell wall of the plant. And what these guys do is then they grind up the leaves.
Right.
And they make an extract, and then they show by EM that there's virus particles in there, and they immunize animals and show protection.
How do they immunize the animal?
They just inject
with all that plant extract too?
Well, it's a good question here. I don't know how much they purify it. Let's take a look. We'll take a look in the materials and methods to see if they do any purification. So they purify the virus-like particles.
Okay.
Now, one cool thing here is that plants don't have sialic acids, right?
Uh-huh.
So the particles will come off the surface without neuraminidase. But these authors say this is a great way to make vaccines. And here's a cool number, Dick. You'll love this. Kilogram of plant leaves, they make 300 doses of vaccine.
Wow.
What do you think of that?
I think it's great. How does that compare to eggs?
Immunization of mice with 2 doses of as little as 0.1 micrograms of purified virus-like particles triggered a strong immune response.
A kilo of plant makes 300 micrograms worth of virus?
300 doses. All right, I can't find it offhand.
Okay.
That's the number I remember. So how much is a kilogram of plant leaves? Is that a lot?
Sounds like a lot.
All right, so 1 egg makes 15 micrograms of flu, which is a dose, which is so for every vaccine shot you get, it's 1 egg.
Oh, really?
So my question is twofold. First of all, is this something that is pharmaceutically feasible to make a vaccine in plants? And secondly, could we make a vertical vaccine farm to make vaccines in your vertical farms?
Sounds like a natural progression of
Can you grow
ideas.
tobacco plants in your vertical farm?
Absolutely.
And we could put this vaccine in them. And
that might not be the only plant that you wanted to use for this.
Absolutely. This is for demonstration. The tobacco is manipulable, and so perhaps you would use a different plant. I know nothing of production.
It's a big leaf.
We have listeners out there who work in the pharmaceutical industry who are involved in bioproduction and scale up. So tell us, is making a viral vaccine in plants something that makes sense? You're going to have to purify it because you don't want any of the plant material around.
But the plant is really easy to grow.
So are cells in culture, and then the virus comes out into the supernatant, right?
Yeah, but you don't need a laboratory to do this.
You don't. It's much cheaper, right?
Yeah.
Dick, do you know of any pharmaceuticals that are made in plants at the moment?
Well, I thought that the hepatitis B protein code was made in bananas. I think they've engineered bananas down at, I think it was Texas Tech, if I'm not mistaken. Texas A&M, I'm sorry, Texas A&M.
I don't know if they actually use that in immunizing people, though.
No, but the idea was there to do that.
Sure.
And I think they've looked at that.
Well, this is a great idea, isn't it?
I think it's a wonderful idea.
And I think putting it together with your vertical farm would be a killer.
I
Because, Dick, you need a lot of plants to make enough vaccine, especially flu vaccine.
That's true.
And if I understand your position, that ain't going to happen on land.
No.
And it would be tough to do even in a GMP facility, a good manufacturing practices facility. How would you have enough area to make all the plants? I think your vertical farm is perfectly suited for this.
I do too.
So let's call this episode the Vertical Vaccine Farm and send a message to the industry that they have to combine your vertical ideas with this wonderful virus-like particle production in
That'd
plants.
Be great.
Okay. And listeners, if you have any ideas, by all means, let us know.
Yep. And that's probably not the only vaccine you could make that way either.
No, there are quite a few others. But, of course, right now everyone's talking about flu, right?
Right.
Okay, I have one more thing to tell everyone before we get to reader email. And this is quite interesting. About 2 weeks ago, Harold Varmus made an announcement. You know who Harold Varmus is,
He's one
right?
of our PNS graduates.
Yeah, he's a
Former
PNS
director
graduate.
of the NIH.
And current director of Memorial Sloan Kettering
And
Cancer Center.
just incidentally, he also has a Nobel Prize.
He has a Nobel Prize.
And he's a nice guy.
He happens to be the co-founder of PLOS,
Oh, right.
Public Library of Science. We quote their papers a lot, which is open access science. He started a website called PLOS Currents, and the first one is called PLOS Currents Influenza. And this is for putting up your papers before they're accepted for publication. So if you want to submit a paper, and you're happy to have everyone see it beforehand, you submit it here.
Interesting.
So he says the key goal is to accelerate scientific discovery by allowing researchers to share their latest findings and ideas immediately with the world's scientific and medical communities. So it uses a website called Google Knoll, KNOL, which stands for knowledge. And you're basically putting up a knoll, a unit of knowledge.
Right.
So, in fact, we have written a paper here on influenza with my colleague Raul Rabadam and Vladimir Trifonov. He's a bioinformatician. We wrote a paper and he put it up there. And you can see it. It's going to be submitted somewhere. And there are many, many papers already on flu. And presumably these will be submitted somewhere. So that's fabulous that you can see this stuff right away. So
what's the
check it out.
drawback to this, Vince?
I'm glad you asked. Dick, when you submit a paper to most journals, the condition is that it cannot have been published anywhere else.
Correct.
Right?
Not only that,
Right.
it has to be reviewed by experts in the field.
Right.
So the facts have to check.
Yeah. So these papers are, as Harold says, these are preliminary because they haven't been
Right.
peer reviewed.
Right.
And he said, but we'll be happy to take them in PLOS. Well, that's fine. But what about Journal of Virology or Parasitology Today?
Yeah.
They're not going to take these papers. So you're right. You're still thinking like an academician, Dick.
Well,
I think
sorry
the vast majority of papers aren't going to go here because
no.
they can't be published anywhere else.
What you might get are rejected papers.
Ah, yes. I certainly have plenty of those.
And that's, I don't know if Harold actually wants that to happen or not.
You know, Dick, there's this model of publishing where you put a paper online and then you have everyone comment on it. And then you could modify it according to the comments over time.
Unless they object to the basic data. Then you have to go back to the lab. Usually that's the basic reason for not accepting a paper, don't you think?
Yeah. They don't believe the data. They don't believe how you interpret it.
Your controls.
Or they want more experiments that you don't have. And they say, well, unless you include these, you're not going to publish the paper.
Exactly.
But, you know, publishing is changing. Scientific publishing is
Yeah.
changing. I wish we had Alan here to talk about this, but this is part of it. And I don't know, I think the journals are going to have to change their ways because more and more scientists are going to want to put their papers up before they're submitted. Raul said in physics they do this all the time.
Yep.
They put up preprints. And in fact, did you know here at Columbia, there is a website where you can put all your preprints there. It's just for the Columbia community to see before they get accepted. So it's not considered being published, but your colleagues can see it. That's kind of good. So
I just got a journal. I subscribe to several now, and one of them is an environmental journal of the Journal of Environmental Chemistry.
Mm-hmm.
And they've gone to publishing in a new format, which I'd like to alert our readers and listeners to. The journal looks like a regular journal with the titles and everything on the front. But if you open it up, you have to read it this way. In other words, you turn the journal 90 degrees. The pages are now wider than they are long. And they've reduced the font size by 2 font sizes.
Oh, my gosh. I can't read that.
Now, the journal used to be this thick. I'm going to about 1.5 inches. Now the journal's about 1/4 inch. They've saved a tremendous amount of paper, but they've made it almost impossible to read. So I think the readers are either going to require that the journal goes back to the way that it used to be or totally electronic.
Do they have an electronic version at this
I
time?
don't know, but I have a feeling that they're moving in that direction by forcing you to object to this way of publishing because I can't read it either. You know, I sit at my breakfast table now with a cup of coffee and I have to use 2 hands to handle the journal, and I can't just sit there and flip pages anymore. I can't look at the graphs any longer. I need a magnifying glass. It's not a good way to do it. It's forcing an old style of publications into a box, which really doesn't fit the situation any longer.
Well, I sit at the breakfast table with my laptop and I read articles.
That's the way to do it.
But it's still a bit bulky. I'd rather have a little tablet that I could do
like
it
a Kindle.
Yeah, like a Kindle, which
like a Kindle.
A little better. I hear Apple's coming out with a tablet.
Really?
Next year. Maybe we can read our papers over coffee on that.
How interesting.
So let's move on to some email. But first, I would like to tell you that this episode of TWiV is supported by Citrix, the makers of GoToMyPC. Now, if you're a frequent traveler for business or even pleasure, you know you still need to keep your business running when you're away, which means you need to have reliable and secure access to your home or work computer. And the easiest way and the most secure way to do that is by using a program called GoToMyPC, which is brought to you by Citrix. And all you need to do this is an internet connection to access your computer from anywhere. It's incredibly easy, an automatic 2-minute setup. You're ready to go in minutes. You go to a website from your computer that you want to share. You click a few buttons, put some information in, you'll get a login ID, and you're ready to go. And then from any internet-connected computer in the world, you can access your data. It's an easy interface to log into. You don't need any hardware, and it's very fast. You can transfer files, you can read your email, you can run any program on your office computer, and it meets stringent security requirements of businesses and government agencies. All the data are protected with AES encryption using 128-bit keys. Pretty
I'm
impressive,
impressed.
huh?
I'm impressed. Very impressed.
So you can try this free for 30 days. You go to gotomypc.com/podcast. That's gotomypc.com/podcast. You'll get a free 30-day trial, and we thank them, of course, for their support of This Week in Virology. Now we have a bunch of email, which is always fun.
Right.
We love when our listeners are involved. And Debbie wrote, I have a question for you, and I thought you would be the best place to ask. There's recent news reporting an outbreak of H1N1 in turkeys in Chile, the exact same virus seen in humans. If this is the case, wouldn't this be a case for concern, given that H5N1 is endemic in the bird population on a global scale? What is the view of the virologists? Dick is pointing to me.
And Vince is pointing to me. No, that's not true.
So, yes, there was an outbreak of H1N1, the same 2009 human pandemic virus that is circulating globally in turkeys in Chile. Yes, it could be an issue. H5N1, of course, is endemic in birds, but apparently it's not yet in Chile. It's in Southeast Asia, China, and other places, Europe. But so, yes, it could be that this H1N1 and the H5N1 recombine. But, there may be already situations where this is happening. So it's theoretically possible. And it's likely that, eventually H1N1 will spread to birds elsewhere, and it'll be in a perfect situation to recombine with H5N1. Am I worried? No, because I think probably there already have been opportunities for these viruses to reassort in the same host that we don't know about. You see, the problem with virology, or maybe it's not a problem, but the property is that there are many, many things that occur that we don't know about. So I think lots of reassorting and co-infections are going on in species we don't know about, and so I'm not particularly concerned. Yes, it's a theoretical possibility, but we don't even know if the reassortants that would form would be transmissible or infectious in humans. So there's so many ifs.
Well, apparently there's a basic difference between the H1N1 and the H5N1 in the sense that H5N1 is a bird-to-bird transmission ordinarily.
Correct.
Whereas H1N1 can be person to
right.
person.
That's right.
So that's the reason why it's crossed down below the border of the equator. But I don't think H5N1 has gone down below the equator. And that might relate to the flyways of the birds themselves rather than because people would ordinarily get very sick from this.
Sure.
So there's an ecology to this that might be worth looking at.
That's a good point. See, the concern, of course, is that the ability to transmit among humans, which, of course, the H1N1 has, could be transferred to the H5, or something could be transferred from H5 into the H1N1 that would make it horribly virulent in people.
That's always
as the H5 is. So, yes, there is a concern. But we knew this would happen eventually, that the two viruses would find a host to co-infect. And as I've said, they've probably done so already. But I'm not particularly worried. I think H5 has been circulating a long time.
Exactly.
It's not transmitting in humans. I think it's had opportunity to reassort with human strains.
Absolutely.
Hasn't yet, so. Which doesn't mean it won't happen, but I'm not terribly concerned.
Yep.
Anthony wrote, Hi, Vincent and Dick. I was listening to TWiV this week and was so excited to hear you talk about flu mist. I work at MedImmune, the company that makes flu mist in California, in an assay development group for flu mist testing. I also wanted to write to say that I love the podcast and listen to it every week while running assays in the lab. Keep up the good work and the great and informative podcast. Thanks, Anthony. Keep that vaccine coming out. Flu mist is the infectious vaccine that you spray into your nose with an inhaler, which I think is great because it mimics a natural infection.
Exactly.
And it's the vaccine of the future. Maybe that and a virus-like particle vaccine will be the future and no more eggs.
Right.
Okay, Judy wrote, I am a high school biology and AP biology teacher, and your podcast has helped me understand a lot and also pointed out that my background is lacking in many areas.
So is ours.
I thank you for both things. It keeps me on my toes. I have a question about CMV, cytomegalovirus.
Exactly. Associated with AIDS.
You know why that is, Dick?
Probably because of interleukin-12 downregulation, but you'll tell me.
All right. So Judy writes, I give blood regularly, and I was originally, 20 years ago, CMV negative, and they would take my blood for newborns. Now I am CMV positive. Can you tell me about CMV and why CMV negative is needed for newborns and how I could become CMV positive and have no symptoms? Okay, that's her first part of the question. CMV, cytomegalovirus, is a herpes virus. It is ubiquitous. Almost everyone is infected either at birth, transplacentally, or shortly thereafter from other kids in schools, very common transmission, so that by the time you're adult, most of us have been infected. But in healthy people, there are no symptoms
Right.
of this infection, and that's why you could be seropositive and not ever have noticed it. It's only when you are immunosuppressed do you have an issue with cytomegalovirus, or largely when you're immunosuppressed, you have an issue with CMV infections. So that's why the HIV-infected people, AIDS patients, have a problem because they're immunosuppressed.
Right. And they're harboring the virus already, so it just comes up.
Exactly. Or if you have an organ transplant, you get chemically immunosuppressed so that you don't reject the transplant, and that allows these viruses to come up and cause disease. So it's actually unusual that she was negative.
Right.
Sorry.
Yeah.
It's unusual that she was negative because most adults are positive, but there could be a few that escape and you were lucky. Now, how did you become positive? You got infected from a kid. If you're a school teacher, it may have been from a
Could have also been a false negative result.
All right. So she actually emailed me a few weeks later and said it was a mistake.
Right.
I'm still negative.
Oh, she's still negative?
She's still negative.
Really?
Yes. She wrote later and said the blood bank called and said they were wrong. The test was negative. So she's still negative. She can still give blood. And she said, why is it important to be CMV negative? Well, you don't want to have the potential of giving the virus to a newborn because you don't know if they're immunosuppressed already, and that would make them very sick. So if for any reason they have to receive blood, it has to be from a CMV negative.
Right.
And other virus negative donor, right? For all the viruses that you test for.
Sure. So there's this test called a torch test.
Oh, what's that?
TORCH. So what do you think the initials stand for, Vince? I know you know this, so we're going to play these games with each other a lot.
Torch.
Torch. It's what you should get when you first discover that you're pregnant. You don't have an idea?
I've never been pregnant.
Well, that's probably the right thing to say at that point. So, okay, I'll just go down the list for you.
Torquemada.
No, not Torquemada. T stands for toxoplasma, actually. So, which is a protozoan infection that you should be worried about catching, because if you do, you've got the potential for infecting your fetus.
That's why when you're pregnant, you should stay away from cats.
Right.
Wow.
So O stands for the orphan virus. R stands for rubella, another virus.
You got to stay away from that if you're pregnant because it causes birth
C,
defects. Cytomegalovirus.
That's right.
And H hepatitis.
That's right.
I got one.
Yeah, well.
Torch. I never heard that acronym before.
Torch test.
Very good. Okay, and then she asks, I seem to remember a CMV promoter used in tissue culture for protein expression. Same one. Yes. Many plasmid vectors have a promoter derived from cytomegalovirus, and that's it. That's the virus you're talking about. Okay, thanks, Judy. Teach those kids high school biology. You know, my high school biology teacher is the reason why I'm in science. Boy, she inspired me.
I had a he.
Do
you remember the name of your teacher?
I do. Barbara Doden.
Barbara Doden.
I don't know if she's still alive, but
mine was Dominic Casuli.
That's right. You mentioned that once, yeah.
Dumont High School.
Barbara was in Allendale, Northern Highlands Regional High School.
Oh, yeah.
Hey, Barbara, you listening? Mrs. Doden.
Please.
Marisano wrote, hello, Twivers. I've been enjoying and learning from your program for some time now. Took a little break to have a go at my preliminaries. One thing that I haven't heard much about and would like to hear more about is attenuation in viruses. And we're going to do a show on attenuation. I think that's a great idea.
What's the best attenuated virus infection that's used as a vaccine that you can think of, Vince?
What do you mean by best?
Well, the one that's been around the longest and the one that's still a live virus vaccine.
Yellow fever.
There you go.
Yeah, that's great. It's a really good one.
It's the best.
And it was the first made in the modern era of virology, I would say.
Yeah.
Yeah, but we'll talk about that at another show. I can't see the screen anymore, Dick.
Uh-oh. Vince is now putting on a second pair of glasses on top of the first pair of glasses.
Oh, that's great.
You know what? You should probably move the screen closer, Vince. It might work better.
Maybe I should move up a little.
All right, we're having an adjustment of the studio here.
A while ago, I read Turner and Chow's revealing paper. Oh, this is very interesting. Prisoner's Dilemma in an RNA virus. I have always felt, incorrectly or not, that this process, high multiplicity of infection, has something to do with attenuation. In light of this, I have wondered why HIV has not become attenuated. I would really appreciate a show discussing viral attenuation, ways in which it occurs, the circumstances under which it is likely, and those under which it is unlikely to occur, telltale signs of incipient attenuation, any pointers concerning an attenuation timescale in the case of recent host switching, etc. Thank you. Oh, that's great. So I looked up this paper, Prisoner's Dilemma. Do you know what the prisoner's dilemma is, Dick?
Whether to escape or not?
It's very interesting.
I can only guess.
Let me tell you what it is very briefly here.
Please.
In a virus infection, if you do a high multiplicity, which means that you put a lot of viruses onto cells, you have a co-infection with two different viruses. And this is somewhat problematic because the viruses don't cooperate, right? They have their own goals. They don't want to cooperate with each other, right?
But there are some viruses called helper viruses, aren't there?
So here we show, yes, that's right. Here we show that the fitness of high multiplicity phage, they're using bacteriophage in this paper, relative to their ancestors, generates a payoff matrix conforming to the prisoner's dilemma strategy of game theory. In this strategy, selfishness evolves despite the greater fitness payoff that would result if all players were to cooperate.
Yeah.
So it's the prisoner's dilemma because a population composed of defectors has a lower fitness than one containing only cooperators. So apparently it is known from game theory strategy, this prisoner's dilemma. So anyway, we will put a link to that, and we will do a show on attenuation. Now, Peter from Korea, who has written to us before, recommended the following article, and it's in Pharmatechnologist, and we'll just mention it briefly. Baxter produces H1N1 vaccine. Baxter has said that its Vero cell production technology, which is mammalian cell culture, has made it possible to complete production of its first commercial batches of H1N1 vaccine 12 weeks after receiving the virus strain, which is very quick. And they're now doing safety testing. So that is another way that some of the new vaccines are being made for the fall in mammalian cell culture as opposed to eggs. David wrote, I recently listened to a podcast about the boundaries of Darwinism. The speaker, John Dupre, talked a little about how viral DNA gets incorporated into host DNA and that this incorporated DNA may make significant contributions to evolution of the host. So he sent me a link to that. This is actually the Darwin College lecture series at the University of Cambridge. It's on iTunes. And in the second term of every academic year since 1986, Darwin College has organized a series of 8 public lectures. Each series is built around a single theme. So this is the boundaries of Darwinism, the first lecture, which we'll put a link to. I was wondering if this would make a good topic for a future TWiV, both from the perspective of describing how viral DNA gets incorporated into host DNA, how, when the incorporated DNA can possibly mutate and alter the host organism. My path to discovering TWiV was an interest in paleontology, which led to an interest in evolution, to genetics, to biotech, to Futures in Biotech podcast, and then to your podcast. Would you call that a tortured route?
Those are a lot of dots to follow here.
So how DNA of the virus gets incorporated into the host, absolutely. We'll tell you about that, and when it can mutate the host. So I have a colleague here, Steve Goff, who knows all about that, and maybe we can talk him into
Wouldn't that be good?
Joining us to tell you about that.
Steve's a very erudite speaker and
absolutely
jocular.
Yes. Tony wrote, an issue came to mind when Dick asked the $64,000 question in TWiV number 43. How many viruses will infect me? Remember that question?
I do.
And you mentioned in the following discussion that probably no more than a few hundred known viruses infect humans. My thoughts linked to the various discussions on humans as an ecological environment and the various bacteria in our skin and in our gut, etc. For example, if I recall correctly, Bonnie Bassler mentioned in a recent TED talk that a typical human intestine will harbor over 200 different types of bacteria. I am currently also reading one of your Pick of the Week books, Riddled with Life, by Marlene Zook, and in this she shared a quote that bacteria make up half of human feces by weight. The role of bacteria in human digestion, especially vitamins, etc., is often discussed. Now, it seems to me that bacteria are good at making all sorts of goodies that assist in their feeding, replication, and defense, but they aren't programmed to be altruistic and generously donate these to the host they live in. I also recall a comment by Craig Venter that for every bacterial population, there is at least one phage that preys on it. So by inference, you could hypothesize that there are probably at least a couple of hundred phage populations busy at work in your gut as well. So given the net material flow rates in the human intestine, aren't we also pretty dependent on the lytic phages that pop those friendly bacteria goodie bags for our guts to pick over? Yet when I browse of the various wiki articles on digestion in the gut, I can't find any discussion of viruses and phages in such digestive processes. There are also various discussions and papers about the impact of antibiotic use on gut flora populations and equilibrium, but wouldn't such swings in food stock have mirrored swings and possibly elimination of the predator phages? Surely the repopulation mechanism for the lytic varieties could prove problematic given our current focus on hygiene and killing 100% of all germs. So finally, my question. Are you aware of any research into the role of phages in the digestion food web? Have any assays been done on these viral components? Do you know the split lytic versus lysogenic phages in this population? Okay, so he's got one other question. Let's do this one. Phages in the gut. Do you know anything about that, Dick? Okay, so there's actually a beginning literature on this. There are some papers that are just starting to come out where people are looking at phages in the gut. So, for example, I found paper in 2008, Viral Diversity and Dynamics in an Infant Gut. And they did metagenomic sequencing of DNA viruses from the feces of an infant. And what they find are mostly phages.
Really?
Right. And these phages did not originate from breast milk or formula.
Huh.
And they change dramatically between the first 1 and 2 weeks of age. And I found another paper where they looked. So in this paper, they're trying to understand Crohn's disease, which is inflammation of the gut, and trying to see if there are good and bad bacteria involved. And they did a metagenomic analysis, and they find, again, a very complicated bacteriophage community associated with the gut mucosa reaching 10 to the 10th phages per cc of tissue. Huge amount of phages.
Wow.
So the question is whether they differ in Crohn's versus normal people, and maybe that plays a role in the disease. And finally, I found a paper called Metagenomic Analysis of an Uncultured Viral Community from Human Feces. This is from 2003, and this is the first analyses from feces, and they found mostly phages again. So there's quite a bit of discovery going on, but the role of these in all the processes that you mentioned, we don't know. No one has done experiments to address that. It's a very interesting question.
Yep.
Really glad that you brought that up. I'm sorry to tell you, we don't know what these phages are doing, but I would be surprised if they didn't play some role in digestion or turnover of material. You know, we're starting to learn that bacteria in the gut are playing a big role in stimulating immunity.
Oh, yes.
So maybe the phages play a role in breaking them up.
Along those lines, there was a recent notice, I guess, on Yahoo News, which I, of course, swear by, or sometimes about the breakthrough in understanding the role of the appendix.
Ah, yes.
And
yes, I saw that. Yeah.
Apparently, the appendix is a safe haven for normal gut flora during times of ablation of gut fauna and flora by, let's say, antibiotics or something else that maybe disaster has happened. And then you can repopulate the entire gut tract's bacterial flora, at least, through the reservoir of this little projection off the side of your gut tract. Now, remember, Vince, I told you I did a lot of work with germ-free animals.
I remember on the whole ride back from Philadelphia.
That's right. So animals without bacteria will obviously not have bacteriophages either. How could they? Right?
Ah, very interesting. How are those animals without bacteria? They get really big ceca, right?
They do. But they can process their food.
Mm-hmm.
They can digest it to a degree. What they can't do is digest anything with cellulose because we don't have an enzyme cellulase. The bacteria do, however. So that's the role of bacteria, is to actually reduce the food bolus down to a reasonable limit so that you can pass it comfortably. I always felt such pain for these poor mice that had this almost granular fecal pellet. It's not even a fecal pellet. It's sterile, and it's like meconium in human beings when we're first born, right? We don't have any bacteria in our bodies at all. What I would like to see is a progressive analysis of the colonization of all tissues in the human over time. I would love to know how that happens.
Sure.
Because, I mean, we have more bacterial cells on us and in us than we do have our own cells.
That's right. That's right. It's huge.
We have more foreign DNA as part of us than we do. So
Yeah.
we're all walking ecosystems and all slightly different. It's an amazing world in and out of there.
It's a symbiosis, right, Dick?
Absolutely. Absolutely.
Okay, he has one more comment. A nitpick on positive polarity RNA. I thought that I had a good grasp on the principles of sense and antisense in DNA coding, but I must admit that you really threw me for a moment when you started talking about positive and negative polarity. The way that I describe it is that I think of a DNA strand as a book where the pages are paired so that writing and mirror writing copies face each other. These equate to sense and antisense. We'll gloss over the fact that sometimes the pairing sometimes flips so that odd pages can contain a mix of page sequences in normal and mirror writing and vice versa. Both senses contain the same information. It's just that the cell's mRNA only reads the sense strand, just like we can only read the normal writing pages in such a book. The plus and minus is just another notational convention for sense and antisense. Surely this doesn't really have anything to do with polarity, chemical, electrical, or otherwise. You're absolutely right. I mean, we speak of antisense and sense. So sense is plus polarity and antisense is negative. It's just a convention. It has nothing to do with chemical polarity, electrical polarity, or any other kinds. I'm sorry.
Political polarity.
But I'm glad you point that political polarity. I'm glad you point that out because there are probably some other people who are confused as well.
By the way, what always confused me about mirror writing, now that he mentioned it, is... Was Leonardo naive enough to believe that people couldn't figure out that if you just held a mirror up to his manuscripts, you could read them?
Can you answer that, Dick?
I can't. I'm posing that as a question. Because he was a brilliant man, and he wrote in mirror writing.
Yeah.
And, you know, it just took a simpleton to figure out that,
I'm
hey, he's
sure.
writing backwards. Look, I can put a mirror up there, and it spells out the word ambulance.
I'm sure some people did.
You
know, Dick, from the beginning of human life, there have been smart humans, right?
Right.
So
I think he was eccentric, and he just enjoyed writing it because he could.
Probably. I know you're like that, Dick.
I can't.
Don't you write your books in the mirror image?
Probably of somebody else's book rather than...
All right, we have a postscript bio-bite, since this seems to be the norm, from Terry. I live in the UK, am 6 months younger than you. How does he know how old I am? I guess we've talked about it, right?
We've revealed ourselves,
not that I mind.
Unfortunately.
And I have been in IT for over 30 years. I recently left a divisional CTO job with one of the big IT players, going into semi-retirement because of ill health. I find virology fascinating because of the many parallels to the theory and techniques of information systems, the sort that makes you ill as well. LOL. Yeah, I got that.
I think viruses are information systems.
It's true. That's why many people in IT are interested in them, right?
Yeah.
I subscribe to a few dozen podcasts to the degree that many just end up filling my iPod with episodes because I don't have time enough to listen to them all. TWiV is one of two where I only have at most one episode, simply because I enjoy their content so much that they don't stay unopened for long. So +1 to all of the other feedback praising comments. Well, that's very nice. Thank you so much, Terry. We love not only praise, but good questions. And I'll post some of those links for you. Matthew writes, TWiV, I was listening to this week's podcast, and I liked your discussion on bats and emerging infectious diseases, along with your guest a few months ago, Eric Donaldson, UNC Chapel Hill. Do you remember Eric?
Absolutely.
And Ed Gates Appalachian Laboratory, we have received funding to use deep sequencing techniques to identify the virome of Western Maryland bats. Once we identify bat viruses, we are going to synthesize them in the lab using synthetic biology techniques, as we did to create the bat coronus parent of SARS coronavirus that Eric published and we talked about, to create those bat viruses and compare them to their human cousins. You were questioning who is looking at bat viruses in the U.S., and it's us.
Okay.
At least at an early preliminary stage. The use of next-generation sequencing, Illumina, 454, etc., to get tens of millions of base pairs in a single sequencing reaction lets us identify the total viral content of a sample, in this case bat guano, rather than looking at known conserved regions of human or animal viruses by individual PCR. I'm sure Eric will let you know how things are going. We should have some real data in a few months. Thanks, Matt. Well, Matt, maybe you'd like to join us on TWiV and let us know what happens, too. Come on when you're ready to talk about it.
Vince, do you think they're going to miss some bat viruses by just looking in guano?
Yeah, I think so.
I do too.
But probably don't want to harm them, right, in this preliminary study.
Well, preliminary study, that's true.
I would think that you're going to miss the respiratory ones, right? Because they can't go through the guano.
And the latent viruses and the integrated viruses.
Yeah. I don't know why they're looking at guano. Matt and Eric, why are you just looking at guano?
I think it's easy to get.
So, Dick, a little parenthetical story. I was at the driving range with my son last night. It was about 9 p.m. It was dark, and the sky was full of bats. And I was like, why are these bats here? It's dark. Usually I only see them at twilight. And my son Aiden said, Dad, there are lots of bugs out. The lights, the bright lights were attracting billions of bugs, and the bats were going bats.
That's right.
It's incredible.
It's a good thing you weren't at the baseball driving range.
Oh, here's our first pun of the episode. There were dozens of bats, quite large, about 8 inches across, brown, and flittering all over, getting those bugs. They're providing a good service, right?
Absolutely. Bats are wonderful. But, the moths have a defense mechanism against this, some of them. They have a radar detection system, and they can pick up on the frequency of the radar, the sonar, I meant to say, and they can avoid the bat. And so some moths are not part of the bat diets because of that. It's an arms race. It's an arms race.
It's fabulous. Dick, the more I listen to you, the more I learn. Gregory,
well, you've got a picture of Darwin sitting on the other screen over there. I just couldn't help but think about that at the point.
I'm going to put pictures up to inspire you from now on. All right, Gregory wrote, Hey, Vincent, Dick, and Alan, great work on TWiV. It has helped me out a lot in deciding for college. I'm a high school senior and couldn't decide between going into pre-med or going into a virology program. To help me decide, I went to iTunes and looked for podcasts on both subjects, and I must say I find virology to be fascinating. I am a fairly smart student and will now apply to Columbia University as it is my first choice. I hear it's a great school for virology.
I think we're being pandered to here.
And I hope I will be accepted. I have a 3.95 GPA and a 27 on my ACT, so I hope I will be good enough to be accepted. In case I'm not accepted to Columbia, do you know any other good schools for virology? Well, Gregory, if you are accepted, I'm starting a virology course next semester. You can take that. Oh, other good virology schools.
But wait a minute, he's a high school student.
Yeah, he wants to go undergrad.
So I don't know any undergraduate schools that allow you to specialize in virology, do you?
No. But there are going to be schools that have a lot of good virologists.
Right.
And so UPenn, Harvard, Stanford are a couple. There is a nice website that I'll post a link to that has a nice review of other schools for virology. But you're right, Dick. You just need to get a good education. But of course, if you end up at Columbia, you can always come and
absolutely
learn from the virologists here.
Yep. By the way,
yeah,
Eric, that was his name, Eric?
His name was Gregory.
Oh, Gregory. Okay, Gregory, I just wanted to inform you something. If you put the weight of money that a physician makes on one scale and the weight of the money that a virologist makes on the other side of the scale, and you just let go, you can hear the noise of the money hitting the bottom of the scale pans from the physician's side of things. So just be aware that while your intellect says be a virologist, your wallet might say something else.
No question about that, Dick.
I hate to say it, but being pragmatic.
But I'm glad that he's decided on virology.
You know what? You can be both.
You could be. You could go to an MD, PhD program.
That's right.
Yeah.
That's right.
But first, go to a good college.
Absolutely.
Patricia wrote, now Patricia was referring to an episode where we said that the rabies vaccine was the only post-exposure vaccine. So Patricia sent a link to an article. Studies conducted in Japan and the United States in the 70s and 80s demonstrated that post-exposure varicella immunization is about 90% effective for preventing varicella if given within 3 days and possibly within 5 days of household or hospital exposure. So chicken pox. So if a kid is in a house and is exposed to someone, you could vaccinate within 3 days of exposure and prevent. So that's another post-exposure vaccine.
Great.
Thank you, Patricia. We always depend on our listeners to correct us, and we have more coming up. Jim wrote, any thought to starting a place to assemble online data from many sources similar to Proteopedia, something to discuss on TWiV? So, there's a big movement now for open science, which means putting all your data online and letting everyone see it. And I have a bunch of links I'm going to post. Open Wetware is one wiki. There was a forum here at Columbia about that. There was a forum at the ASM general meeting last spring. And a lot of people want to do this. There are obstacles, just like there are obstacles in open access publishing. So, it's a great idea. It's been discussed extensively, and its time will come. Jesper wrote, Dear fellows of TWiV, it is my understanding that us humans live in peace and symbiosis with some bacteria. Is there any such arrangements with any virus? Isn't that a great question? Oh, they're all great. Another way to phrase the question, if all viruses were to be removed from the world, would we be better off? That's great. Here's a follow-up, and we'll answer that. A follow-up, sure. Even more abstracted from the lab bench. If all viruses were gone, is it reasonable to believe that new ones would come into existence? How fast? From where? In one of the TWiV episodes, someone said any sufficiently complex systems has parasites.
Yes.
So I assume given time, something is bound to fill the niche of viruses.
Hear, hear.
So are the viruses help us, Dick?
Well, when you say us, I hope you're not just referring to our daily lives as human beings. So I would refer back to an episode we had on the ocean and viruses and the trading of genes between various microorganisms in the thin film that lies on the top of the water. The viruses do a horizontal transfer of genes from one organism to another. It means that they don't have to be sexually compatible or even have sex in order to trade genes.
Sure.
So without viruses, you're going to miss that huge exchange program. It's a genomic exchange program, right? So you take the viruses away, and now you're going to really slow down.
So exchange among bacteria, gene exchange, right?
Or perhaps even algae species, too, right?
Now, the other thing about the ocean viruses is they turn over the carbon.
Right.
There's so much lysis of bacteria.
That's right.
It actually impacts the carbon cycle and the geothermal cycles. Huge.
Exactly.
So if you took all that away, you'd screw up the whole world's ecology.
Exactly.
Right? Now let's look at mammals. So we have endogenous retroviruses in almost all mammals. They're there for some reason. They're still there. We don't understand why, but they must have a reason.
Are all those intronic sequences of viral origin?
Not all the introns, no. But a good percentage of all the extra sequence, everything that differentiates us from a chimp, for example, is mainly retroviral sequence.
Right.
So it must be there for a reason, or else it would be gone.
Are there? Okay.
Right? We have, as you have pointed out many times, we get infected, and so do animals, with viruses and don't have any disease.
Right.
There must be a reason for that. So the thing is, Dick, if life had evolved without viruses, it probably would evolve in a way to compensate for their absence. But if you took them all away now, if you could suddenly turn a switch and get rid of all the viruses from the globe, what do you think would happen?
Well, I think immediate benefit would be less human suffering.
Right. Would there be any negative effects, as he said?
The gene exchange program that's in vogue right now would stop.
Right.
And we don't have a clue as to how that figures into the maintenance of ecological process.
I think we'd have a massive ecological disaster.
I think so too.
And it would impact evolution of species as well.
I think that is part of evolution of species.
So I mean, is it true to say that anything that evolves along with us becomes a part of the whole fabric and it becomes essential in some ways? So viruses have evolved with us. We've learned to take advantage or to use their presence.
The ecologists have a word for differentiating all of the species from some of the important species. Mm-hmm. The term is keystone.
Ah.
Right, and the keystone is that wedge-shaped center brick that went into an arch that the Romans invented.
Right.
So if you take that block away, the whole thing falls down. I think also if you take the ones along the side, the whole thing falls down. But we have systematically reduced the biodiversity of most ecological settings through disturbances and encroachment. And there is a point at which there's a tipping point, I think. It's like the rivet theory of the airplane. It loses rivet 1, nothing happens. Rivet 2, nothing happens. Rivet 3, rivet 26, bam, the wing falls off.
Right.
Nothing happened up to rivet number 26. All of a sudden, the last rivet goes, and there everything goes. So maybe the viruses would affect life the same way. We don't know, and we hope we never find out.
I wish we could do that experiment, right?
Maybe in a microcosm you could.
His other question is, if you took all the viruses away, would new ones come into existence?
That's interesting.
So here's my take. I think no, because we haven't seen any new viruses for many years. They only come out of animals. Those are the new viruses that we discover.
Oh, you mean in people?
So I think they evolved very early on when life was first evolving, and I don't think the conditions are the same any longer for them to emerge. So that's why we don't see any new ones, any completely new viruses emerging. I mean, everything that comes out is something that's in an animal already and nothing totally new. Do you know what I mean, Dick?
I do know what you mean, but I would counter with the argument that we don't even know how many species of things there are on the planet. And so until we have a complete list, and we will never have a complete list, by the way, but until we have, let's say, a fairly good idea about the breadth and depth of biodiversity, we can't say whether everything does or does not have a viral association. So you expect to see new viruses the moment you discover a new species.
Right.
Right?
But those are pre-existing. Those are pre-existing viruses. No new ones are just emerging as the first viruses emerged.
You mean spontaneous production of viruses from nothing?
Well, it's not going to be from nothing, but
exactly.
It has to be.
So what about all these variations on H1N1, H5N1, all those H3N2s? Those are variations on a theme. Each one can be considered a new virus, but you know where the parent strains are, and I agree with you that these are minor changes in a basic plan. What you're looking for is something completely radical, like the Andromeda strain or something.
Something totally different. We don't see anything new.
Yeah.
It's just versions on a theme, as you say.
Right.
And that's why I don't think that even if you took all the viruses away, no new ones would emerge.
I betcha I know where there are brand new ones.
Where?
In Lake Vostok.
Where's that?
It's about a mile below the ice sheet in Antarctica. It's the size of Lake Ontario.
Is there life there?
We don't know that yet. We have a probe. The Russians have a probe that's about 3/4 of the way through the ice, and they're figuring out how to go into the lake without contaminating the lake so they can find out if there is life there.
Okay.
That ice sheet has been there for millions and millions and millions of years, geographically and everything else isolated. If there is life in Lake Vostok, it's likely to harbor some brand new things that we've never seen before. So maybe these geographically isolated regions have something that will surprise you, Vince.
I don't doubt it, but it's just because we haven't found them. But I don't think any brand new ones are evolving.
Has Craig Venter's ocean explorations turned up anything startling?
Yeah, it turned up viruses that we'd never known about, genes that we'd never known about before. But that's just because we failed to find them.
Oh, well, isn't that what his question is?
His question is, if you took away all the viruses,
would
new ones emerge?
Spontaneously.
Yeah, well, spontaneously, or however they emerged originally. That's the question. We don't know, right?
Well, if viruses are obligate parasites, then they had to evolve after their host cells appeared. If that's the case, then there does appear to be some credence to the hypothesis that there is some spontaneity to the evolution of viruses as opposed to other forms of life.
So why haven't we seen this happening lately, though?
Maybe all the niches are filled.
Okay.
I don't know.
Well, those are the issues anyway.
These are dumb answers from a now emeritus professor.
No, it has nothing to do with it. Doesn't mean you're dumb because you're emeritus.
These are wild guesses. How's that?
All right. We have a couple more, and then we'll wrap this up. Blake writes, great program. I love the show and currently do some undergrad research in virology, and it has captured my interest. I'm applying to graduate school and have attended some fairs, but I'm still struggling to find a program that's right for me. I wanted your opinion on schools that had good virology or microimmunology PhD programs. I know this is a broad question, but I figured someone with your experience could point me in the right direction. Mm-hmm. So I think here at Columbia, UPenn, Harvard, Stanford, Baylor, Wisconsin are great virology programs, to name a few. There's great immunology at Yale, Scripps, Emory, NYU, UCSF. So I'll put, actually, there's a great listing at virology.net on grad programs in virology. I'll put that up. Mm-hmm. And also, of course, we talked about it.
And some wonderful European schools also.
Yeah, I mean, I don't mean to be comprehensive at all. The great European schools, yeah.
Yep.
And of course, I don't want to slight anyone by not mentioning them, so go check out virology.net.
Yep.
John wrote, I just finished listening to episode 44, and during the podcast, there was a question about any post-infection vaccines being available besides rabies. I recalled a couple of articles indicating that research into post-infection vaccines for Ebola and Marburg virus has been ongoing for some time, and apparently an experimental vaccine has been used on a person at least once, although it was not clear that the lab worker actually was infected. You remember in Germany there was a infection or thought to be an infection, a woman in a lab, and they gave her post-infection experimental vaccine. We talked about that.
Yeah.
You mentioned the time for the immune system to respond to a vaccine was typically 7 to 21 days. Given the speed that these viruses act in humans, I'm curious how these post-infection vaccines would generate a rapid enough immune response to provide protection. Thank you again, and thanks to Dick and Alan for taking the time to share your knowledge in an entertaining and informative way. I enjoy your podcast very much and look forward to each week's episode. So yes, there are some experimental post-infection. These Marburg Ebola vaccines are mainly for post-exposure, I would think, in laboratory accidents or in Africa when someone gets infected, you would give it to them.
By the way, it's not a new concept because during the early days of exploration of sleeping sickness, which is a protozoan infection caused by a trypanosome, some of the first victims that made their way back to England were treated with isolates of trypanosomes that they actually drew from the patient's blood, grew them up, then ground them up and injected them back into the patient in hopes of stemming the flow of this infection without realizing, of course, that they were undergoing an antigenic variation type of thing. So that's the reason why it didn't work, but at least they were thinking about post-infection vaccination then. That was back in the
very good.
turn of the century.
How is it that you have enough time? Well, rabies, as we discussed, takes a while to travel up the nerves. If you're bitten far enough away from the CNS, you have 2 weeks. But some of these hemorrhagic fevers have a shorter incubation period. I think the vaccines induce some innate responses early on, and that keeps the virus replication in check until adaptive responses can kick in.
That's right.
Right, so that may be part of the reason why they do work. But you do have a time window. You don't have forever. Andrew wrote, Hi, guys. Just a couple of quick comments regarding TWiV 44 and fruit bats following your conversation. First, contrary to Dick's statement, many bat species definitely do migrate. These are mainly species that live in more temperate regions, but even the tropical species may travel to many hundreds of kilometers, probably in search of food. The bats in which Ebola, Marburg, Hendra, and Nipah have been found belong to the family Pteropodidae. I don't know how to pronounce it, Dick. Pteropodidae, or Old World fruit bats. These bats are not found in North or South America, and hence those viruses are unlikely to be found in bats in the U.S.
I think what I was driving at was that there's no seasonal migration of bats like there is for other waterfowl, or like the caribou or something like this. I'm sorry that I misled you in the statement that said that they don't fly far.
Okay.
They do. They go far.
Also, fruit bats do eat the food that is still on trees, and not so much the fruit that has fallen to the ground.
Okay.
And RE TWiV 45 and the option of culling wildlife species to control disease. One main problem with the concept is that it is unlikely to be possible to entirely wipe out a population as you suggested, but it is also unlikely to be carried out for ethical reasons, as well as culling part of a population tends to lead to increased migration of animals into the culled area and hence potentially bringing in more disease. See the perturbation hypothesis for badgers and TB in the UK. And also, culling part of a population will probably lead to an increased reproductive rate in the remaining animals, improving the condition for infectious diseases. And finally, many wildlife species perform important services in the environment, also as you suggested, e.g. fruit bats are important pollinators of forest plants, and they disperse seeds, hence playing a role in maintaining the forests where they live, i.e. get rid of the fruit bats and you'll get rid of the forests.
Right.
Yeah, those are all really good points.
By the way, I'm sorry.
Go ahead.
But in the old days, when it was known as Rhodesia, they actually slaughtered animals along the border of Rhodesia and the country north of that, which I think is Zambia. I'm not sure. Or Mozambique. I'm not sure which country it is. We'd have to look at a map. But they were killing tons of animals at the border to prevent the introduction of sleeping sickness into the cattle that were being grown just south of the border in Rhodesia. When Rhodesia finally gained its independence and became Zimbabwe, they stopped doing that, and interestingly enough, they've now got huge problems of sleeping sickness in Zimbabwe.
All right, so
We're looking for Zimbabwe now, which used to be Rhodesia, and the country north of it.
Zimbabwe is south of Zambia.
Zambia. Okay, fine, I was right then. Zambia.
And to the west of Mozambique.
Yeah, and so it became tradition in that country when it was Rhodesia to actually kill the animals at the border to prevent them from bringing their trypanosomes into the country, which was cruel and stupid. But you can see what happens when they stopped.
Yeah. Otherwise, great work. I really enjoy listening in. P.S. Peter Dasak works for the Wildlife Trust and was previously head of the Consortium for Conservation Medicine, not World Wildlife Fund Federation.
Sorry about that,
Dick.
too.
You feeling okay, Dick?
My heart's rating a little up there.
So Andrew gave us a couple of suggestions for next shows. One is an article about a new vaginal gel for women that blocks HIV. So these are very interesting. We'll get some experts to tell us about the vaginal gel. It's a very interesting new way of preventing HIV. So thanks, Andrew. Now, Russ sent us a link to an article called Osteoporosis Drugs Are Effective in Killing Flu Viruses. Now, this is just an article. Well, I went to the article in Reuters, and it gave me that nasty ad.
Yeah, a little pop-up.
2 existing drugs used to treat osteoporosis may be effective in killing influenza viruses, researchers in Hong Kong have found. Now, first of all, you don't kill viruses because they're not living to begin with, right, Dick?
Right.
And there are 2 drugs.
That much I do know.
And this is what I find difficult because this isn't published, so I can't tell what they're doing. In their experiment, the researchers exposed human cells that had been infected with the influenza virus to the 2 drugs. They observed that the drugs triggered extra production of a type of white blood cell called gamma delta T cells, which went on to kill human cells that were infected with the virus. So this doesn't make any sense because they say they expose human cells in culture to virus. Where are the gamma delta T cells coming from? It doesn't make any sense. And I can't find this in the literature. So I'm not sure what that means. And if it's never published, then it probably isn't correct.
Right.
But anyway, thanks, Russ. Russ also sent us a link to an article, Vaccinia Infection After a Lab Accident. So a man at CDC was working with Vaccinia virus, and apparently he got an eye infection, which was quite serious. And then he was treated for a long time with antibiotics because they didn't know what it was. But then finally they realized it was Vaccinia, and they gave him acyclovir. And he recovered and went back to work. And these Vaccinia viruses, which are usually thought to be benign, can infect and cause serious symptoms in some people. Absolutely. So Russ writes, acyclovir works on thymidine kinase, which is a viral enzyme, and is usually used for herpetic infections. I did not know that it could be used to treat pox virus. Would it work for polio? Does polio and Vaccinia use a viral polymerase for replication? Well, it works for pox virus because pox viruses also encode a thymidine kinase enzyme, which is important to phosphorylate the drug once it's in a cell, and then it becomes active. It's an inhibitor of polymerases. Polio would not be susceptible because it's an RNA virus, but Vaccinia, yes, all viruses need polymerases for replication. Some of them encode them, and others use cellular polymerases. And we're going to talk about that in Virology 101, right, Dick?
We will.
All right, we'll wrap it up, Dick. Let me see if there's another one. One more.
Okay, one more.
Jim wrote, I thought you guys might enjoy a look at the real 3D structure of bacteriophage T4 and its life cycle. So Jim, who works for a company called Sayet, sent a link to some movies of T4 replication. So we'll post that so that you can see it. And now that should do it. We have lots more email, but Dick needs to leave. So I'm going to end up with my pick of the week, which is a website called BioNumbers, a database of useful biological numbers. So if you need to know how many ribosomes there are in a cell, how many cilia there are on the surface of a cell, etc., you go there and you will find it. How many trypanosomes are in your intestine?
How many chloroplasts are in
Exactly.
Wow.
Anything you want to share with our audience this week? You have reading a book on science lately, Dick?
I'm reading lots of books on science.
Name one.
Just one. The latest book on science that I read is a book co-authored by Sylvia Earle,
Mm-hmm.
and it's called An Atlas of the Oceans.
Oh, cool.
And it's fabulous. It's absolutely wonderful.
We will make that your pick of the week. Thank you, Dick.
Yep.
Dick can be found. Dick, thanks very much for joining me today. I appreciate it. I have a wonderful conversation with you always.
Vince, I wouldn't miss it. I'm just a little bit late today, that's all.
I'll let you go. Dick is at Racaniello.org or verticalfarm.org.
.com. Or medicalecology.org.
Excellent. We're at TWiV.tv. You can send your questions and comments to TWiV at TWiV.tv. Thanks for listening, everyone. We'll be back next week. Another TWiV is viral.